Welcome, everyone, wherever you're joining us from, to today's session of Global IMMUNOTALKS, the series that brings all aspects of immunology to a global audience. And today we welcome Isaac Chiu. Isaac Chiu is an associate professor of immunology at Harvard Medical School. In fact, he has carried out most of his training at Harvard, doing a PhD first with Michael Carroll, and then staying in Harvard for postdocs, initially with Tom Maniatis, and then later on with Clifford Wolf. And then in 2014, he started his own lab at Harvard Medical School, where he joins us from today. Now, Isaac's research focuses on the interactions between the nervous and the immune systems, and his particular interest is in how neurons sense and respond to microbial signals. His lab has been doing very exciting work uncovering mechanisms by which microbes can actually activate sensory neurons, for example, through secreted toxins and proteases, and how these interactions can actually lead to conditions such as pain and itch, and also how these interactions with microbes can then allow neurons to convey information to cells of the immune system. And I think a key goal of Isaac's research is to define mechanisms that underlie interactions and that allow perhaps one to develop better treatments for pain and inflammation. In addition to also exploring how innate immune molecules such as gasdermins contribute to things like neurodegeneration in the central nervous system, which is a long-standing interest of his. Now, as usual, today's talk will be followed by a Q&A session in Twitter or X, in which Isaac will endeavor to answer your questions over the next couple of weeks, and instructions for how to ask questions will be put on the screen at the very end of the talk. So in a minute, I'm going to ask Isaac to deliver his lecture, but as is usual in this series, I'm going to ask Isaac first a personal question or a couple of personal questions before the talk. And those are, what strategies, Isaac, do you use to stay innovative and motivated in your research? And what role has serendipity played in your scientific discoveries?
Thank you so much, Caetano. It's my pleasure to be here for the Global ImmunoTalks. Thank you for those questions. I think that innovation and creativity are two of the driving forces for all scientists. For me, I use several strategies. One is to try to not be limited just to my field or my topic. So I try to attend meetings and conferences and talks outside potentially my immediate field, and really learn something, the cutting edge technologies or tools, and also interacting with scientists, diverse scientists with different approaches. The second way I think to be keeping innovative is also to really recruit a dynamic team. So as a PI, I have postdoctoral fellows and graduate students coming from neuroscience, immunology, microbiology, and we, brainstorm ideas and really having the different perspectives. I think some of the most exciting science is happening at the interface of fields. So having neurobiologists and microbiologists getting together, we often spark really exciting new ideas. And this keeps me motivated and excited to see also their passion. And then that feeds into the second question about serendipity. So just as an example, when I was a postdoctoral fellow in Clifford Wolf's lab, I was new to neuroscience, and Clifford is a famous neuroscientist who studies pain. And I just worked with a fellow postdoc who taught me neuroscience. And I said, okay, how do pain fibers and microbes interact? And we were just playing around with our mutual knowledge. I added bacteria to nerves, and he had, calcium imaging, and we discovered that the neurons were sensing bacteria. So this curiosity-driven approach with very serendipitous discovery really changed the course of my career, because that's really now a big part of what we study is how do sensory neurons directly sense microbes? But it wasn't a planned, discovery. And I think that also illustrates that other thing is to go after maybe unexplained findings and really allow creativity and exploration in the lab.
Well, yes, chance favors the prepared mind, and the broader your horizons, the more your mind is prepared. So,
Yes.
on that note, thank you, Isaac, for those words. And I'm going to ask you to start sharing your screen and deliver your lecture, which is entitled Sensory Neurons and Their Roles in Barrier Immunity. So,
Yes.
Isaac Chiu.
Thank you so much. Today I'm going to talk about neuroimmune interactions at the barriers. And these are actually some images of sensory neurons in different barriers, such as the skin and the meninges and the gut. And I'm going to hope to convince you that they play an integral role in host defense and immunity. So my talk today, this is a brief outline. First, I'm going to introduce pain, itch, and also neuroimmune interactions. And then I will dive into two of these key barrier sites where we think these neuroimmune interactions play a role. One is the first is the skin, and I'll tell you about how we discovered that Staph aureus, a bacteria, activates nerves to drive itch, and then this induces significant inflammation because of the itch. Secondly, I'm going to focus on the gut, a densely innervated organ where we think neuroimmune interactions is very important. And recently we found a role for TRPV1 neurons in modulating Tregs in the gut. So firstly, for the introduction, as I mentioned, all barrier sites are actually densely innervated by sensory neurons. And that's because one of the key roles of these neurons are to interface with the environment and detect stimuli from the outside world. That includes mechanical, thermal, chemical sensory functions, as well as detecting microbes, which are another stimuli that we receive. And we think also coordinate physiology at these barriers, whether it's with immune cells or epithelial cells. You can see again the beautiful network of sensory fibers in the skin, the lungs, and the gut. So our question is, how do they interact with the immune system during homeostasis and during inflammation? So sensory neurons mediate protective reflexes. So just like the immune system, one of their key roles is also to defend us from potentially harmful stimuli. And two of these reflexes are illustrated here. One is pain and one is itch. So pain, as you know, when you touch something potentially harmful, for example, a noxious heat source, this will cause an immediate response, unpleasant sensation that leads to a motor withdrawal reflex. And itch is also another reflex where it's thought that you can remove something potentially harmful from the surface of the skin. These kind of reflexes are nociceptive, and actually Sir Charles Sherrington, one of the great fathers of modern neuroscience, coined this term nociceptor to describe the pain reflex. Interestingly, in terms of immunology and inflammation, pain is also one of the four classic cardinal signs of inflammation as defined by Celsus, the Roman physician from 2,000 years ago. So we know that pain accompanies inflammation. Well, now we're trying to appreciate that the pain fibers, the nociceptors that mediate pain, also directly modulate or mediate inflammation itself. So that's an exciting question, is the role of neurons in mediating the inflammatory response. So these neurons that are called nociceptors reside in the peripheral nervous system. So their cell bodies are actually in the dorsal root ganglia, which are right outside of lining the spinal cord. And they send one peripheral axon, which terminates in peripheral tissues like the barrier sites, where they detect potentially noxious stimuli. And then this action potential is transduced via their central axons to the spinal cord, where then the signals relay to the brain and it's perceived as pain. So what are some of the things that these neurons are tuned to detect? Well, we know there are specific ion channels and receptors on the nerve terminals of those receptors that detect noxious temperatures, such as extreme heat or cold. They also have ion channels like TRPA1, which detects noxious chemicals. And also they have mechanoreceptors to detect injury and also ATP and DAMPs released during injury. What we're also finding is, like the immune system, these nociceptors can also detect molecules from pathogens, which is another major organismic danger. And so over the years, our lab and other labs have defined some of these mechanisms by which these neurons directly sense pathogens. So my lab found, for example, that Strep pyogenes and Staph aureus, 2 gram-positive bacterial pathogens, directly activate nociceptors via pore-forming toxins like streptolysin S and alpha-hemolysin. These pore-forming molecules are highly damaging, and they cause pain by inducing ionic influx through through the nerve terminals, and then you get a firing of action potentials. Other labs have now also found that some of these neurons express TLRs like TLR4 and TLR5, which can detect gram-negative, LPS, and flagellin. And these are also gated to TRP channels like TRPA1 and TRPV1. There are beyond bacteria, there's also evidence from Dan Kaplan's and others that, for example, Candida albicans or fungal pathogens can also directly activate nociceptors. So this is a growing area of interest of how the sensory system is tuned to pathogens. The other really interesting thing is that these neurons not only signal to the brain to mediate pain perception, they also signal locally in the tissues to mediate immunity. And this is a term called neurogenic inflammation. So besides this action potential going up to the CNS, there's also back propagation of action potentials along the neighboring branches of the neuron, which leads to local neuropeptide release. So there are neuropeptides stored in the nerve terminals, such as substance P and CGRP. These immediately upon pain perception gets released, and then they can act on immune cells. So this is just highlighting a couple of examples. For example, mast cells have a receptor called MRGP-BR2, which is gated directly by substance P. And this causes mast cell degranulation. So you get an immediate pain-induced neurogenic inflammation caused by mast cells. Substance P and CGRP actually also may have opposing roles in terms of macrophage function. So, for example, substance P may induce pro-inflammatory cytokines, whereas CGRP, acting through its receptor complex formed by RAMP1 and calc-RL, actually has been shown to suppress TNF and NF-kappa B production and also induce IL-10. And this might be actually involved in, for example, wound healing responses, where it polarizes the macrophage response. So besides talking to immune cells, and neurons, microbes and immune cells, these neurons we think is integrated into the overall host defense response. So again, the questions of defining how neurons sense microbes and how they crosstalk with immune cells is a big question. I didn't have a slide to illustrate this, but the neurons also directly sense cytokines and immune mediators. So there's a tri-interaction going on when you have infection and host defense. And there's a growing body of work from multiple labs showing that nociceptors play critical roles in these barrier sites. So the skin is particularly well studied because the types of sensory neurons in the skin are better defined and it's a little bit easier to approach. So very elegant work from multiple labs, including Dan Kaplan's lab, Nico Gaudenzio's lab, and Uli von Andrian's lab, have looked at how nociceptors regulate multiple dendritic immune cell types like dendritic cells, mast cells, and also downstream T cell responses. We recently found that the meninges, which is another barrier, this time an internal barrier that surrounds the brain, you could see the beautiful nociceptors. These neurons talk to macrophages in the meninges during meningitis and brain infection. And also, we and others are working on nociceptors in the airways that mediate lung inflammation. And I'm going to focus today also on the gut, which is another key barrier site. But there are other barriers where there are lots of pain fibers, such as the cornea, which also might mediate these interactions. And finally, recent work actually from a lab in China showed that nociceptors regulate splenic germinal center responses and antibody production. So this is a very exciting area of research. So now I'm going to switch to the very first story that I'm going to focus on, which is in the skin. And here I'm going to focus on this first interaction I mentioned. How do microbes talk to sensory neurons? And how does this then regulate inflammation. So this work was carried out by a very talented postdoc, Liwen Deng, in my lab, and actually originally was started by Kimbria Blake, who is a former PhD student in my group, in close collaboration with Alex Horswell's lab at University of Colorado and Flavia Costa in his lab. And we have other collaborators here. So this is about itch, and itch is just very briefly also to talk about itch. So itch is something that evokes, by definition, a desire to scratch. It's the kind of annoying, unpleasant sensation. And you can see these pictures of multiple types of mammals which itch. So it seems to be a conserved reflex response. So why do we itch? I think that's actually a philosophical question that potentially some even itch biologists don't exactly know the answer to it, but there are some possibilities of how this is protective. So one is to remove skin irritants on the skin that could be harmful to us. Another is to remove ectoparasites like mosquitoes. But also we know that itch induces an allergic immune response, and also there's a feedback loop where allergies induce itch. Finally, there's a response or reflex in the brain called mirror itch, where if you see other people scratching, you actually have neural circuits that might mediate copy behavior. So could this also be somehow protective in the setting of removing of things on the skin? So we wanted to ask a question that has not really been answered much, is what about the impact of microbes on itch? So we know multiple exogenous stimuli like mosquito bites and also certain plants have exogenous chemicals that can induce itch. However, we know the skin is constantly exposed to different microbial pathogens, and also there's a commensal microbiome, but very little has been known on whether microbes on the skin impact itch-mediating neurons and whether this can impact also pathology. So itch fibers or pure-receptive fibers are cousins of nociceptors. They also reside in the dorsal root ganglia. And like I said before, they take signals from the skin in this case to the brain and mediate an itch sensation. So we were curious also because of the potential connection of microbes to chronic itch diseases. So atopic dermatitis, or AD, is a chronic skin disease characterized by eczematous skin lesions and chronic itch. So in fact, there's a part of this disease which is known as the itch-scratch cycle, which you scratch until you feel relief from the itch, but this exacerbates skin damage, and then you get another itchy cycle. So this is thought to be actually fundamental to pathology of atopic dermatitis. AD is actually quite common. It's in 10% of adults and 20% of children. And another key characteristic of AD is barrier dysfunction and microbial dysbiosis. In particular, Staphylococcus aureus, which is a gram-positive human bacterial pathogen, is found on over 90% of the itchy lesions of atopic dermatitis. So if you find, normally Staph aureus is actually not on a normal skin, it's more in the nares of the nose. But on atopic dermatitis patients, you will find Staph aureus on the skin, and especially in the AD lesions. So this led to a hypothesis, could they be producing something or directly inducing itch in some way? Now, a lot of people have been studying the relationship between Staph aureus and skin inflammation in the context of AD, and it's known that Staph makes many different virulence factors, including toxins like phenosoluble modulins. Also, they make superantigens that can drive T cell responses, and also proteases, which can damage and induce barrier destruction. So are any of these factors also involved in itch? So to answer that question, we set up a mouse model to study staph exposure and the effects on itch and inflammation. So we adapted a widely used model of epicutaneous exposure. So here we basically shaved and depilated the back skin of mice, then topically applied a methicillin-resistant Staph aureus strain, USA 300, onto the back. And then we occluded it with a tegaderm, which allowed the bacteria to then invade the skin. And over the course of 5 days, you see significant inflammation. caused by this exposure. So you can see thickening of the epidermis and also infiltration of immune cells that is characteristic of atopic dermatitis lesions. So our question is this accompanied by itch? And so we in our lab use neurobehavioral assays to study itch. So one is to actually record spontaneous scratching behavior using this infrared behavioral box. So they're in the dark, so they're more comfortable to do behaviors like scratching. And we're looking down on top of mice, some of which have been exposed by Staph aureus. You could see they're scratching the site of exposure or around the site. And we quantify that over the course of 90 minutes. And we could see that MRSA exposure causes significant induction of spontaneous itch in both females and male mice. The second way that we measure itch is called allonesis. So this is touch-induced itch. So you might imagine when you have like a surface that's inducing touch, like a itchy sweater, this could trigger itch by just rubbing against these lesions. So how do we test that in mice? What we do is we actually poke the animals with a filament, in this case, that causes touch. So a very thin filament with 0.07 grams pressure. Normally, the mouse will not respond, but if there's inflammation or sensitization, they will now induce scratching. So we test this in 9 times, pokes on the back. And what you could see here is that Staph aureus exposure causes these mice to start responding a lot more to this touch. So it induces allokinesis. So is this scratching actually relevant to pathology? So Li Wen actually made a very interesting observation that when she removed the gauze and allowed the mice to scratch, there was a lot of inflamed skin after a few hours that was not only at the site of exposure to the bacteria, but also all around the back. So the mice are scratching, and the scratching, then she could quantify the degree of damaged skin. And what then she did was either wrap the mice with a bandage shown here, or do like a pedicure, like, cut their nails. And in both cases, what she found was that if you prevented the mice from scratching their back, there was a significantly less damaged, inflamed skin on the animals. So suggesting that the behavior of scratching itself is a significant cause of skin damage. So again, this itch-scratch cycle is important to think about for dermatology. So what mediates this itch? Well, we noticed when we're looking at the nerve fibers of the skin, here we're using a fluorescent reporter strain, NAV1.8, NAV1.8 is a voltage-gated sodium channel that's highly expressed in sensory fibers. So we have a TD tomato reporter. And then what we're doing here is exposing these mice to Staph aureus that's labeled in green with GFP. And so we could see close juxtaposition in the epidermis of nerve fibers with bacteria, which suggested that potentially there are direct interactions of the bacteria with the neurons. And so we hypothesized maybe these bacteria are producing factors that could directly activate the itch fibers. And so to test this hypothesis, we teamed up with a really expert microbiologist, Alex Horswell, who has multiple mutant strains of Staph aureus, which target different aspects of their virulence. So one of their key virulence mechanisms is this AGR quorum sensing mechanism, where quorum sensing peptides activates the induction of multiple virulence factors, including proteases and toxins. And what we found is if we ablate AGR, this induction of spontaneous itch that occurs with wild-type Staph aureus over PBS control is now abolished. So we think something downstream of AGR is controlling the itch behavior. So we next tested whether there's a role for alpha hemolysin or PSMs. Now, alpha hemolysin is the factor we had previously found Staph aureus can release to activate pain fibers when you have subcutaneous infection. But neither of these mutants played a role in the itch. As you can see, they're the same as wild type. So another major class of virulence factors controlled by AGR are proteases. In fact, Staph makes 10 different extracellular proteases. And in collaboration again with Alex, he had a mutant strain in which he knocked out all 10 proteases. And in this case, we found that again, wild type bacteria caused significant itch over a control, but the delta protease mutant no longer was able to induce itch. So What we did is, and I won't show you the data, that we actually then systematically targeted all, each of these different proteases that out of the 10 to narrow it down to which one might be mediating this phenotype. And it turns out that the key one is one of the proteases called V8, which is encoded by the SSPA gene. So here's some of the data to show that using a specific isogenic mutant strain for SSPA, that could significantly decrease the spontaneous itch compared to wild type staph. And then you could restore this by complementation on the chromosome with SSPA. So again, these are strains from Alex's lab. And this also was the case with the touch-induced the allokinesis. So you could see the SSPA mutant has much less touch-induced itch, but then the restoration by the SSPA complementation. What about the skin pathology or dermatitis? So another way that we can measure the amount of damage to the skin is by using a skin score, which is a combination of different measurements, erythema and swelling, and also water loss measurements. And you could see again that this V8 protease critically mediates skin damage as well in dermatitis, with decrease there with the mutant and restoration with the complementation strain. Another measure is transepidermal water loss, which is a measure of epithelial barrier function. So again, we think that this protease can mediate damage as well to the barrier. And this is, you could evidenced here. So this V8 protease seems to be critical for driving both itch and also skin damage. What about its relevance to atopic dermatitis? So Richard Gallo, who's a dermatologist and expert on atopic dermatitis at UCSD, helped us by looking at actually lesional skin samples from atopic dermatitis versus non-lesional skin versus healthy controls. And what he found was that the mRNA transcript for SSPA is significantly induced in the AD lesional skin. So what is V8 protease? V8 protease is actually a serine protease produced by Staph aureus. It's a core part of its genome. Previous work has shown it plays an important role in different aspects of host microbe interactions. For example, it induces biofilm dispersal and also inhibits complement by cleaving complement components. So this can allow the bacteria to evade the immune system. And recent work, a proteomic study showed that actually quite a lot of blood proteins in the host is cleaved by this V8 protease. So we asked, can this protease induce itch as well? So what we did here is we injected recombinant V8 protease into the mice. So here we're injecting them actually into the cheek. And you can distinguish between itch versus pain behaviors by measuring scratching, which indicates itch, versus forepaw wiping, which indicates pain. What you can see is V8 protease can induce very potent scratching itch behavior, actually even as much or more than histamine, which is the prototypic itch inducer. And then on the other hand, it does not induce this wiping pain behavior, whereas capsaicin, which is the active ingredient in chili peppers that causes pain, can induce pain but not itch. So V8 is sufficient to drive itch but not pain. Can it activate sensory neurons that mediate itch? So here what we're doing is we're culturing dorsal root ganglia sensory neurons and doing calcium imaging. And this allows us to look at responses of subsets of neurons to different ligands. When we added V8 protease, we could see subsets of neurons responding by calcium influx. And some of these also responded to things like histamine or capsaicin. So there are different subsets that respond to certain ligands. And one of the things we noticed was that V8 responsive neurons many of them can also respond to other itch ligands like histamine, chloroquine, and S1P. So this means that pruritigenic neurons can respond to this specific protease. So how is it responding? So one of our hypotheses is that potentially these neurons are detecting these proteases with specific receptors called PARs. So proteinase activated receptors are G protein coupled receptors, which have a tethered N-terminal ligand. And what happens is when the protease cleaves that N-terminus, it exposes the ligand, and then it actually self-activates itself. So teaming up with Rithwik Ramachandran, who's an expert on PARs, we sent him V8 protease, and he has a luciferase-based assay where you can look for potential cleavage of different PAR receptors. There's PAR 1 through 4, what we found is that V8 protease very nicely cleaves PAR1, but not PAR2. So PAR1 and PAR2 are actually protease receptors that have been linked to itch in the past by sensing exogenous proteases and also endogenous proteases, such as those from mast cells. So this is interesting to us. So we now focused our study on PAR1. So what we did is we obtained PAR1 knockout mice as well as neurons. So F2R is the gene encoding PAR1. And what you could see here is that when we look in this calcium imaging assay, PAR1 knockout neurons, this is actually work by Natalie Vigneault, which is a collaborator, no longer was able to induce neuronal activation when you have PAR1 deficient neurons. So is this playing a role in vivo? What we did here is, because PAR1 is actually expressed by multiple types of cells, we obtained a conditional knockout strain, F2R flox, crossed it with TRPV1 So TRPV1 is an ion channel expressed by sensory neurons, including itch and pain fibers. So this now will ablate F2R just from TRPV1 neurons. And then we performed the Staph aureus exposure experiment. What you could see here is that TRPV1 conditional knockout mice, PAR1 conditional knockout mice no longer show spontaneous itch induction compared to control mice upon staph exposure, and also no longer show itch induced by touch, so allokinesis. And this also has important implications. So can we then target PAR1 to block itch, right? So there's actually an FDA-approved drug called Vorapaxar, or trade name is Zontivity, that's been used to block PAR1 to treat clotting disorders. So we asked, what if we gave animals V8 protease and Vorapaxar? You could see that Vorapaxar at different doses can significantly decrease the itch caused by V8 protease. So then Lewin asked, what if we give mice Vorapaxar? So during Staph aureus exposure. So what she did was orally gavage Vorapaxar into mice daily before and after exposure to Staph aureus. And this also significantly decreased both spontaneous itch and allokinesis after staph exposure. Importantly, it also significantly decreased this itch-induced skin damage. So you could see recording here that Vorapaxar significantly also decreased the amount of inflamed skin. So here, what I'm showing you here is a bacterial virulence factor V8 protease, which is secreted on the surface of the skin, which induces significant itching, and this itching then induces scratching and skin damage. So particularly, it acts on this receptor PAR1, which is expressed on sensory neurons. And this behavior actually induces the inflammation. And then this probably feeds then into a scratch-induced itch scratch cycle and further damage. And we can block PAR1 using either genetic or pharmacological approaches, and this prevents staph-induced itch and skin damage. So this is really the first illustration that I wanted to give you. Now I'm going to turn to a different place where we think sensory neurons play a key role in barrier immunity. So this is now the gut. And I'm going to highlight one story that was a very close collaboration between my lab and Dr. Christophe Benoit's lab in my department. And it was carried out by two very talented postdoctoral fellows, so Yang Zhu in Christophe's lab and Kim Mearshart in my lab. Kim is now a research staff scientist at the Mayo Clinic, and also in collaboration with Diane Mathis, Steve Liberles, and other people at Harvard. So this is just switching gears. Earlier, I told you about the skin. Now we're looking at the gut. And we know that there are many diverse types of neurons that innervate the gut, both anatomically and molecularly. So this is just an image of the neuroanatomy of different parts of the gut. So there are vagus nerve as well as sensory neurons that innervate from the spinal cord, and also sympathetic neurons. So it's very complex. And also there's a complex molecular and cellular diversity of neurons in the gut. And that, I mentioned these extrinsic neurons like vagal or nodose ganglia or dorsal root ganglia neurons, but also there's intrinsic neurons, the enteric nervous system, which in the gut itself, there are dozens of different types of neurons. So it does introduce a question of how do we map neuroimmune interactions in the gut when there's such a complex system. Um, just a note before I get into the story, we did show a couple of years ago, together with David Artis in back-to-back papers, that these neurons also crosstalk with the gut epithelial cells. So we found that pain fibers specifically talk to goblet cells in the gut to modulate mucus production. So there's multiple neural immune and neural epithelial crosstalk going on in the gut. So in terms of the neuroimmune crosstalk, this is a review article that we recently wrote just to highlight the complexities of this neuroimmune communication. So there are multiple extrinsic and intrinsic neurons in the gut that produce also multiple kind of molecular mediators, whether it's neuropeptides like CGRP and substance P, like I mentioned, but also neurotransmitters. And then multiple types of immune cells in the gut, like ILCs, dendritic cells, mast cells, and macrophages, can respond in different ways to each of these neuronal mediators. So a big question in the field is how do distinct neuron subtypes cross with immune cells? So Christoph's lab and I wanted to tackle this question using a specific approach called chemogenetics. So chemogenetics is a way to temporally and specifically control the activity of different neurons. So how it works is that you express a specific receptor called a DREAD. Now, the DREAD is an engineered receptor that doesn't see normal endogenous ligands, but there's a synthetic ligand called CNO, which will activate the DREAD. And what we do is you can express the DREAD in a Cre-dependent manner. So here, what we did is we actually used a virus strategy, an adeno-associated virus, which will only target peripheral neurons. So that includes all the neurons in the extrinsic DRG and nodose ganglia, as well as the enteric nervous system. But then we infect only positive cells that can drive this expression. So by combining the AAVs with the DREAD and also specific Cre lines, then we can target the DREAD just to those neuronal subsets. And then we can activate them with a CNO. So here we're using the activating DREAD. And then what we did is we just systematically look at the changes in the gut immune system. So teaming up again with Christoph's lab, we performed detailed flow cytometry of the gut using 28 different markers. What we decided to do was activate 8 different lines for 14 days to look at kind of, over the course of 2 weeks, what does neuronal activation do to the immune system? So first, to validate our approach, there is an M-cherry protein, which is also expressed by this viral strategy that is co-expressed with the DREAD. So we could see M-cherry labeling in the types of neurons we might expect given these 8 different Cre lines. So here we've used TRPV1 Cre, which I'm going to focus more on, but also piezo2, MRGD, and others that target the enteric nervous systems like CHAT, VIP, and NOS. And you could see labeling in different ways, patterns in the nodose ganglia, the dorsal ganglia, and the myenteric plexus of the gut. So then what happens when we activate all of these subsets and look at the immune system in the gut? So this is a heat map which kind of captures all of the significant changes. So we looked at all of them, but these are the ones that stood out as actually robustly and significantly changed. So just as an example, MRGD neuron activation causes increased levels of these mononuclear phagocytes in the ileum. Just to highlight 4 of these changes, CHAT positive neuron activation suppresses neutrophil numbers in the ileum, whereas NOS positive neurons actually induce Th17-like RO gamma positive T cells in the ileum. And we also, just to point out, we looked at 3 different parts of the gut, the ileum, cecum, and colon. So there are different interactions going on, I think, in a specific way in each parts of the gut. I'm going to focus on the discovery we made on here. So TRPV1 neurons, which are actually nociceptors, had the largest change. You could see actually inducing multiple changes in different immune cell populations in the gut. And particularly, we focused on the downregulation of RO gamma T cells in the gut, Tregs in the gut. So Tregs, as many of you know, are critical regulators of tissue inflammation. In the gut, they play key roles in oral tolerance, and also regulation of responses to the microbiota. So there are two major flavors, the Helios positive and RO gamma positive Tregs, and they both play roles in oral tolerance and tissue inflammation. So it's very interesting for us to find a specific interaction of neurons with these RO gamma Tregs. So this is just to show you the data. So this is, for example, flow cytometry data from these 8 different Cre lines for specifically colonic RO gamma Tregs. You could see when we activate the TRPV1 specific subset causes downregulation of these Tregs. And then we also looked in these mice, which we're now calling TRPV1 ADC mice, compared to controls, whether this is specific to the colon or what about in other tissues. So there is also a significant downregulation in the spleen, but there wasn't a huge change in mesenteric lymph node or lungs or ileum. So in the gut itself, it seems focused on the cecum and the colon. So what about the kinetics of this? Does it take 2 weeks for this to occur? So we looked at different time points after this neuronal activation, and it does indeed take some time. So by day 13, you get a significant downregulation of Tregs over time. So how is this occurring? Well, one of the things that we notice is that it, in fact, is regulating proliferation. So looking at Ki67 levels within different immune subsets after the TRPV1 neuron activation, we see that, in particular, the Tregs show decreased Ki67 levels, both total RO gamma and Helios positive Tregs. Another thing that could be occurring is actually regulation of immune cell trafficking. So here we use this Kaida mouse, which allows photoconversion of cells from the red to the green. So you can track the exit of cells from the gut to other tissues. And so what we found is that TRPV1 neuron activation caused actually significant fold change and indicating exit of cells, not only Tregs, but also a bunch of other types of immune cells from the gut. So there could be a combination of these mechanisms that's occurring. I won't dive into the data, but we also did single-cell RNA-seq and transcriptional profiling of the Tregs, and we found also transcriptional changes in the Tregs after neuronal activation that indicate potential mechanisms as well. So what are the consequences of this TRPV1 Treg crosstalk? So what we did is we first activated the neurons and then exposed these mice, in this case, to DSS colitis. So what you could see here is that following neuronal activation, the mice that were activated had significantly more weight loss following a DSS exposure than the control. And also this was accompanied by worse histological damage. In addition, we also exposed these mice to Citrobacter rodentium after neuronal activation, and Citrobacter it's an effacing bacteria of the gut that causes inflammation. In our case, we found slightly worse weight loss in the TRPV1 ADC mice, but also significant spread of the bacteria out from the gut to the spleen and the liver that was not there in the control mice. So it's possible that one mechanism is that the loss of Tregs leads to this increased inflammation and susceptibility to Citrobacter infection. Which neurons mediate this phenotype? So I mentioned that there are actually 2 subsets of neurons that express TRPV1. One is the dorsal ganglia neurons that mediate pain that I talked about earlier, and the other one are vagal neurons that mediate actually gut to brain signaling through the brainstem. So to answer this question, what we did is we actually directly injected the virus into the spinal cord, which only labeled then TRPV1 neurons in the DRG, but not the nodose ganglia. And then we did the neuronal activation. And you could see that this DRG-specific activation was able to recapitulate the total neuron activation. So we think the DRG neurons are critical. We did the same for the nodose ganglia, but did not see this phenotype. And we also targeted DRG neurons by ablating them and showed that the phenotype disappears. So we think specifically these spinal DRG neurons that mediate pain are the ones that cause this downregulation of Tregs. So, but what is the molecular mechanism of this So I mentioned that the neurons release neuropeptides, and neuropeptides are stored in the vesicles of the nerve terminals. But we thought, could these neuropeptides be talking to these Tregs, for example? So what we did here is we crossed this TRPV1 ADC mice to the CGRP knockout, so calc-A knockout, or substance P knockout. This is TAC, which is encoded by the TAC1 gene. You could see when we knocked out calc-A and did the neuronal activation, the Treg downregulation no longer occurs in the colon, but this is not affected by substance P. So we think CGRP is involved. So how does CGRP work? Well, it acts on a co-receptor complex formed by RAMP1 and calc-RL, which is a GPCR that signals via adenylyl cyclase. So we asked, could RAMP1 be involved? And so actually, when looking at single-cell RNA-seq data of colonic Tregs, these RO gamma Tregs that are especially affected by our activation had higher levels of RAMP1. So this suggests that maybe the CGRP is acting through RAMP1 on the Tregs. So by imaging, we could also see instances where CGRP nerve fibers is close to FOXP3 positive Tregs. So what we did is we then bred a RAMP1 conditional knockout strain flox mouse with a FOXP3 Cree and then TRPV1 ADC mice. And what you could see here is when you ablate RAMP1 in the Tregs, this abolishes also the ability of the TRPV1 neuron activation to induce suppression of Tregs. So this suggests that one of the mechanisms could be CGRP released by TRPV1 neurons acting directly on RAMP1 and Tregs to induce this suppression. So I'm going to summarize now my talk. So what I've shown you here is that sensory neurons play an important role in inflammation and host defense. So they can directly sense microbes and their products to induce pain and itch. And also they can signal to immune cells. I gave you one example of Tregs. And this could then have consequences for inflammation. So in the skin, Staph aureus directly activates itch fibers via protease, and this drives scratch-induced skin damage. And in the gut, our lab and Christoph's lab performed this chemogenetic screen to find new neuroimmune interactions. So by activating different subtypes of neurons, we revealed one very interesting crosstalk, which is that TRPV1 neurons, which mediate pain, actually over the course of time suppress gut Tregs through the neuropeptide CGRP and also the receptor RAMP1. And this then probably leads to increased inflammation when you have challenge to the gut. It's interesting philosophically why pain would suppress Tregs. Maybe the presence of danger or damaging molecules in the gut, maybe you want to then shut down the brakes in order to induce an immune response. But generally, I think there's lots of open questions of how these neurons crosstalk with microbes and immune cells, and hopefully our discoveries could eventually lead to actual therapies for itch, for inflammation, and other neurological and immune diseases. So this work would not have been possible without an amazing lab. I mentioned, our diverse team of people from different perspectives, neuroscientists, immunologists, microbiologists. I highlighted work from Li Wen in my lab, as well as Kim, and also collaborators with Christoph, Diane, and Yang Yang. But there's so many other very talented people who have been in my lab, who are alumni now, and also who are currently in my group. And I also want to thank my funding sources and other collaborators. So happy to take questions, and thank you for all of your time.
Well, thank you very much, Isaac, for this fascinating talk and sort of demonstrating this intimate interplay between these sensory neurons and the immune system, as well as their ability to respond directly to microbial challenges and perhaps other challenges. This is clearly a very interesting area, and also one that we will see a lot more of, I suspect, in the future. So just to remind the audience that questions for Isaac Chiu's talk should be asked via X or Twitter. You've got the instructions up on the screen. And over the next few days or weeks, Isaac will endeavor to answer all of your questions. And so it just really remains for me to thank Isaac Chiu for his wonderful lecture today, and thank all of you who log in from all around the world. Thank you for your support, and I hope you will continue to support the Global IMMUNOTALKS initiative. A reminder that next week's lecture will be given by Monica Walkers, so please tune in next Wednesday. But for now, wherever you happen to be in the world, have a great rest of the day, and thank you again for your support.
I'm a huge fan, so thank you for having me on the Global Immunotalks.
Thank you, Isaac. Bye, everyone.
Bye, everybody.